In this video, we will talk about column chromatography. Chromatography is the term used to describe a separation technique in which a mobile phase carrying a mixture is caused to move in contact with selectively absorbent stationary phase. There are a number of different kinds of chromatography which different in the mobile phase and the stationary phase used. Uh, Column chromatography was first developed by American chemist TTD in 1900 and MS Twist the Polish partners in 1906 used to use absorption column and investigation of plant pigments. Here is basically the history of column chromatography in which the American chemist TTD in 1900 described first this method. He take a mixture of plants component in a column and then these components are separated into different pigments or colors. Column chromatography is one of the most useful methods for separation and purification of both solid and liquid. This is solid liquid technique in which stationary phase is solid and mobile phase is liquid. Now, what is the principle of column chromatography? It is based on adsorption. Mixture of the component dissolved in mobile phase introduced into the column components move depending upon their relative affinities. Adsorbent column chromatography, the adsorbent picked in a glass column. Here is a glass column in which we pick the adsorbent. Uh, Uh, the solvent, the mobile phase that moves slowly through the packed column. A solvent used is a mobile phase called the eluent. Here is the solvent which is used as mobile phase, and this is called the eluent, and the mixture move slowly through that packed column. Here is the basic instrumentation and diagram of all the component of column chromatography. You can see it here. Uh, A compound attracted more strongly by the mobile phase will move rapidly through the column and the eluent form or come up. The column dissolve in the eluent. In contrast, a compound more strongly attracted to the stationary phase will move slowly through the column. It is very simple. Experimental aspects of column chromatography. Adsorbent. The usual adsorbent employed in a column chromatography are silica, alumina, calcium carbonate, calcium phosphate, magnesia, starch, uh, etc. This is the adsorbent which is used in column chromatography. Alumina is generally suitable for chromatography of less polar compound. Silica gel gives good result with compound containing polar functional group. Alumina is used for compound which is less polar, and silica gel give best result for the compound which is contain polar functional groups. Adsorbent and column chromatography should meet following criteria. Here are some basics characteristics which should be followed by the adsorbent which is used in column chromatography. Uh, Particles should be spherical in shape and uniform in size. Second one is the mechanical stability must be high. They should not react chemically. It should be useful for separating for wide variety of compound. It should be freely available and inexpensive. The particle size of the commercially available grade is in the range of 50 to 200 micrometer. Selection of stationary pairs. Success of chromatography depend upon the proper selection of stationary pairs. It depends on the following. Uh, if we measure that how the successful is our technique, it depend upon the proper selection of stationary pairs. Here are the some factor by which it depend. uh, Removal of impurities, we should remove all the impurities from the stationary phase. Number of components to be separated, length of the column used, affinity difference between components, and quantity of the adsorbent used. Mobile phase. The mobile phase act is solvent uh, developed for eluent. The function of mobile phase are as developing agent to introduce the mixture into the column is solvent to developing agent. To remove favor component of the column is a eluent. Uh, the choice of solvent is depend on the solubility and characteristics of the mixture. The solvent should also have sufficiently low boiling point to permit ready recovery of eluted material. However, polarity is seen the most important factor is in absorption chromatography. Different mobile phase used an increasing order of polarity. Here is the different mobile phases used in terms of their increasing order of polarity in which petroleum ether, carbon tetrachloride, cyclohexane ether, acetate, benzene, toluene, acetate water, in which petroleum ether is the least polar, or we can say it is the non-polar, and water is the most polar component. It can be used either up in paper form or a mixture of solvents. Column characteristic. The main function of all column is to support the stationary phase. The material of the column is mostly good quality neutral glass since it should not be affected by solvents. An ordinary Bertie can also be used as a column for separation. Column dimension length and diameter ratio 10 ratio 1. It is length and 30 ratio 1 diameter and 100 ratio 1. Various accessories are attached to the top of the bottom uh, of the column for the maintenance of the elution process. The length of the column depends upon uh, number of components to be separated, types of adsorbent used, quantity of the sample, affinity of the compound toward the adsorbent used. Better separation will be obtained with a long, narrow column than short, thick column because the number of plates will be more. A preparation of column. It consists of glass tube with bottom portion of column picked with glass wool, cotton or wool may be contained is best fed above which adsorbent is packed. After packing, a paper disc kept on the top so that the adsorbent layer is not distributed during the introduction of sample or mobile phase. Here is glass wool to prevent the stationary phase of being washed out of the column, and the column packed with silica gel or alumina completely standard with solvent, with solvent. Layer of solvent on the top of column. Uh, Baking techniques in column chromatography. There are two types of preparing the column. They are dry baking, dry or dry pelling, or wet baking or wet pelling. The column should be free from impurity before using column. It should be washed properly and dry it. Before filling column with stationary paste, cotton, glass, wool is kept. It should be uniformly filled. Dry packing technique. Adsorbent is packed in the column in dry form. Fill the solvent till equilibrium is reached. Uh, Demerit or air bubbles are entrapped between mobile page and stationary page. Cracks appear in the adsorbent layer. After filling, trapping can be done to remove wide spaces. Here is column chromatography technique. You can see here in which solvent is added to it. Uh, Water packing technique, ideal common technique. The material is slurred with the solvent and generally it is added to the column end portion. Solid stationary phase settle uniformly and no crack in the column of adsorbent. Solid settle down while the solvent remain up while this solvent is removed. Then again cotton plug is placed. Now it is time introduction to the sample. The sample, which is usually a mixture of component and is dissolved in a minimum quantity of the mobile phase. The entire sample is introduced into the column at once and get reserved on the top of the portion of the column. From this zone, individual sample can be separated by a process of dilution. Here is the basic experiment which is shown in this figure, in which different colors are shown in this column. We can separate this color separately, like first of all we separate the yellow color in one tube and then the next green color in another tube. uh, Column chromatography whole process is discussed here, in which keep adding new solvent, challenge the baker want the yellow stain to drop, then each of the them solvent go down, and then is separated. Uh, Development technique elution. By elution technique, the individual components are separated out from the column. The two techniques are isocartic elution technique. In this elution technique, some solvent composition or solvent of the same polarity is used throughout the process of separation. Example is chloroform only. Gradient elution technique. Gradient mean gradually. Solvent up gradually increases polarity or increases elution strength are used during the process of separation. For example, initially benzene, then chloroform, and then ethyl acetate, then chloroform. Detection of components. How we detect different components of the sample. If the components separated in column chromatography procedure are colored, the paragraphs of the separation can simply be monitored visually. If the compound to be isolated from the column chromatography are colorless, in this case small fraction of the element are collected sequentially in the labeled tubes and then in the composition of each fraction is analyzed by TLC. Eluting the sample components A, B, and C separate is a column process in which these components are separated. A fraction can be collected in test tube while beakers. Each color are separated in different test tube. Analyzing the fraction, analyze the fraction by 10-layer chromatography. Factor affecting column efficiency. The first factor is dimension of the column. Column efficiency has been improved by increasing length weight ratio of the column. Particle size of the column packing is the second factor which affect the column efficiency. Separation to be improved by decreasing the particle size of the adsorbent. Number third, activity of the adsorbent. Fourth, temperature of the column. The speed of the elution increase at higher temperature. Fifth one is the packing of the column. Sixth, quality of solvent. Solvent having low viscosity is giving better results. Now the application of column chromatography. The first application is separation of mixture of compounds. Second one, it is used in the purification process. It is also used in isolation of active constituents. Estimation of drugs and formulation. Isolation of active constituents. Determination of primary and secondary glycosides in digital leaf. Separation of diastereomer is in this in separation of diastereomer column chromatography is also used. Now the advantages of column chromatography. Any type of mixture can be separated. Any quantity of mixture can be separated. Wider choice of mobile phase can be used. Automation uh, is possible. Now, there is some disadvantages of column chromatography too. Time consuming, more amount of mobile pages required. Automation make the technique more complicated and expensive. Thank you for watching. See you in next video.